Hi everyone, in this video I am going to explain you about M phase or dividing phase of cell cycle. Under this specially prophase and metaphase. Division phase or M phase or mitotic phase of cell cycle. Division phase or M phase lasts for only about an 1 hour in 24 hours duration of cell cycle of a human cell. The M phase represents the phase when the actual cell division or mitosis occurs. In animals, mitotic cell division is restricted or only seen in diploid somatic cell, except in some social insects. Against this, the plants can show mitotic division in both haploid and diploid cells. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. M phase or mitotic phase requires only 1 hour of 24 hours of cell cycle in human cell. M phase represents the phase which is called actual cell division or dividing phase. Generally, in animals, mitotic cell division is occurred in diploid somatic cells except in some social insects. Against this, in plants, both haploid and diploid cells undergoes mitotic divisions. Mitotic division phase is the most dramatic period of cell cycle, involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cells is the same, thus M phase or mitotic phase is also called equational division. Generally, the number of chromosomes is same in parent and daughter cells in mitosis. That's why mitosis is also called equational division. For convenience, mitosis has been divided into 4 stages of nuclear division. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. The M phase starts with nuclear division corresponding to the separation of daughter chromosomes, which is called karyokinesis, and usually ends with the division of cytoplasm, which is called cytokinesis. The number of chromosomes in parental cell and daughter cells are same. Due to that, mitosis is also called equational division because parental cell and daughter cells are identical. For our convenience, mitosis has been divided into 4 stages of nuclear division, and M phase starts with nuclear division or karyokinesis and ends with cytoplasmic division or cytokinesis. Mitosis done in 2 steps, karyokinesis and cytokinesis. Mitosis karyokinesis is divided into 4 following steps, such as number 1 prophase, number 2 metaphase, number 3 anaphase, number 4 telophase. In mitosis, particularly in very first step, karyokinesis or nuclear division done in 4 different steps, such as prophase, metaphase, anaphase and telophase. Very first one, prophase. Prophase, which is the first step of karyokinesis of mitosis, follows the S phase and G2 phases of interphase. In the S and G2 phases, the new DNA molecules formed are not distinct but intertwined. Prophase is the phase which is first stage of karyokinesis of mitosis, followed by the S phase and G2 phase of interphase. In the S and G2 phases, the new DNA molecules formed which are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material, which means chromosomal condensation first started in prophase of karyokinesis of mitosis. The chromosomal material becomes untangled during the process of chromatin condensation. The centriole, which had undergone duplication during S phase of interphase, now begins to move towards opposite poles of this cell, anastral and amphiastral mitosis. In higher plants, centrioles are absent and no asters are formed, which is called anastral mitosis. Simply, mitosis without asters is known as anastral mitosis. But in case of animals, the asters are present and the mitosis is described as amphiastral or astral mitosis. Centriole duplication occurred in S phase of interphase. The duplicated centrioles now begins to move towards opposite poles of the cell. Here mitosis is broadly classified into two types based on centrioles and aster rays: anastral mitosis and amphiastral or astral mitosis. Anastral mitosis generally seen in higher plants. Because centrioles are absent in higher plants, the mitosis without aster rays is known as anastral mitosis. In animals, the asters are present and they helps in cell division. This type of mitosis is called amphiastral or astral mitosis. The completion of prophase can thus be marked by the following characteristic events. Chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Centrosome, which had undergone duplication during interphase, begins to move towards opposite poles of this cell. Each centrosome radiates out microtubules called asters. The completion of prophase can thus be marked by the following characteristic events: chromosomal condensation, chromosome composed of two chromatids which are attached together at centromere. Centrosome, which had undergone duplication during interphase, begins to move towards opposite poles of the cell. Each centrosome radiates out microtubules called asters or aster rays. The two asters together with spindle fibers forms mitotic apparatus. Cell at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus and nuclear envelope. Here, asters form spindle fibers and disappearance of Golgi complex, endoplasmic reticulum, nucleolus and nuclear envelope occurred. Let's see the diagrammatic representation of early prophase and late prophase. In early prophase, the size of nucleus increased, chromatin material starts condensation. In late prophase, cell organelles are disintegrated or disappear. Nucleus also, which means nuclear membrane also ready to disintegrate. Chromosomes or chromonova fibers are formed by the condensation of chromatin material. Let's check these diagrams carefully for your better understanding. Second one, metaphase. The complete disintegration of nuclear envelope marks the start of second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. Metaphase starts with complete disintegration of nuclear envelope or nuclear membrane. By this stage, condensation of chromosomes is completed and they can be observed clearly under the microscope. Here chromosomes are completely condensed and this is the best phase to study, or this is the best phase to see chromosomes in cell cycle. This is the stage at which morphology of chromosomes is most easily studied. At this stage, metaphase chromosome is made up of two sister chromatids which are held together by the centromere. small disc-shaped structures at the surface of centromere called kinetochores. Metaphase is the best phase to study morphology of chromosome. At this stage, the chromosome is made up of two sister chromatids which held together by the centromere. Small disc-shaped structures at surface of the centromere called kinetochores. These structures serve as the sites of attachment of spindle fibers formed by the microtubules to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole and its sister chromatid connected by its kinetochore to spindle fibers from the opposite poles. The plane of alignment of these chromosomes at metaphase is referred to as metaphase plate. The structure serves as the sites of attachment of spindle fibers is kinetochores. The metaphase is characterized by all chromosomes coming to lie at equator, which is called metaphasic plate. The plane of alignment of chromosomes at metaphase is referred to as metaphase plate. Chromosomal fibers, discontinuous kinetochore which runs from pole to centromere, and supporting fibers, continuous non-kinetochore which run from pole to pole. Arranged in cell, centromere lies at equator and arms of chromosomes remain directed towards poles. Here chromosomal fibers are continuous and discontinuous. Discontinuous are kinetochore which run from pole to centromere. Continuous are non-kinetochore which runs from pole to pole. The key features of metaphase: spindle fibers attached to kinetochores of chromosome. Chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fibers to both poles. Key features are spindle fibers attached to kinetochores. Chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fibers to both poles. Let's see the diagrammatic representation of transition to metaphase. See, here chromosomes are completely condensed, nuclear envelope completely disintegrated, centrioles forms asters, asters forms spindle fibers. These spindle fibers are attached to kinetochores of chromosomes. After attaching, all chromosomes are aligned, or chromosomes are laid in a plane in the center of cell, which is called metaphase plate or equatorial plate. Let's check these diagrams carefully for your better understanding. Up to here, under M phase, particularly under karyokinesis, prophase and